Biotechnology has entered a phase where humanity is no longer merely studying life, but actively rewriting it. For thousands of years, evolution operated without conscious direction. Mutations occurred randomly, natural selection filtered outcomes, and biological change unfolded slowly across generations. Now, within a single laboratory cycle, scientists can modify the genetic blueprint of an organism with precision measured at the molecular scale. At the center of this revolution stands CRISPR, a gene editing system that has transformed genetics from a descriptive science into an engineering discipline. Alongside it, breakthroughs in epigenetics, telomere biology, and mRNA technology are reshaping how we think about aging, disease, and even the possibility of human enhancement. The ethical tension is unavoidable. Are we curing disease, or are we redesigning humanity? CRISPR did not originate as a human invention in the conventional sense. It was discovered as part of a bacterial immune defense system. Bacteria, constantly attacked by viruses, evolved a mechanism to capture fragments of viral DNA and store them within their own genome. These stored sequences act as molecular memory. When the virus attacks again, a protein guided by that stored sequence recognizes and cuts the viral DNA, neutralizing the threat. Scientists realized that this programmable cutting system could be adapted for targeted gene editing. The enzyme most commonly used, known as Cas9, functions like molecular scissors. Guided by a short RNA sequence designed in a laboratory, Cas9 can locate a specific DNA segment and cut it with extraordinary precision. Once the DNA strand is cut, the cell's natural repair machinery activates. Researchers can exploit this repair process to disable genes, correct mutations, or insert new genetic sequences. The elegance of CRISPR lies in its simplicity compared to previous gene editing technologies. It is faster, cheaper, and more accessible, lowering the barrier to genetic experimentation. Laboratories across the United States and globally have adopted CRISPR as a standard tool, accelerating research in cancer therapy, inherited disorders, and infectious disease resistance. The potential to remove genetic diseases before birth has intensified debate. Conditions caused by single gene mutations, such as certain blood disorders, are prime candidates for CRISPR-based correction. Clinical trials have already demonstrated success in modifying blood stem cells to treat severe inherited conditions. The scientific logic is straightforward. If a disease arises from a known genetic mutation, correcting that mutation at its source eliminates the pathology. However, when editing occurs in embryos, the modification becomes heritable. This means future generations inherit the engineered change. The line between therapy and enhancement becomes blurred. Aging represents one of the most compelling targets of gene editing research. Aging is not a single process, but a collection of biological mechanisms including DNA damage accumulation, mitochondrial dysfunction, cellular senescence, and telomere shortening. Telomeres are protective caps at the ends of chromosomes. Each time a cell divides, telomeres shorten slightly. Eventually, they become too short to protect the chromosome, triggering cellular aging or apoptosis. Some researchers are investigating whether manipulating telomere maintenance pathways could delay aging. Telomerase, an enzyme that extends telomeres, is active in stem cells and certain immune cells. Reactivating telomerase in aging tissues may extend cellular lifespan, but uncontrolled activation is associated with cancer risk. The biology is delicate. Extending life at the cellular level may increase oncogenic potential. Epigenetics adds another layer of complexity. While DNA sequence provides the genetic blueprint, epigenetic modifications regulate how genes are expressed. Chemical tags attached to DNA or histone proteins influence whether genes are turned on or off. Environmental factors, stress, diet, and toxins can alter epigenetic patterns. Unlike genetic mutations, epigenetic changes do not alter the underlying sequence but can profoundly influence phenotype. Emerging research suggests that some aspects of aging are driven by epigenetic drift, gradual changes in gene regulation patterns over time. Scientists are exploring whether resetting epigenetic markers could rejuvenate cells without altering DNA sequence. Experiments in animal models have demonstrated partial reversal of age-related markers, fueling speculation about biological rejuvenation therapies. The biohacking movement has amplified public interest. Outside traditional academic institutions, independent researchers and entrepreneurs experiment with genetic modification, supplements, and self-directed longevity protocols. While some biohacking efforts focus on quantified self-tracking and lifestyle optimization, others venture into controversial territory, including do-it-yourself gene editing kits. Regulatory frameworks struggle to keep pace with democratized biotechnology. The ethical concern is not merely safety but equity. If gene enhancement becomes viable, access may be limited to those with financial resources, potentially widening social disparities. The question of designer babies encapsulates these tensions. Pre-implantation genetic testing already allows screening embryos for certain genetic conditions during in vitro fertilization. CRISPR extends this capability from selection to direct modification. Traits influenced by multiple genes, such as intelligence or athletic performance, are far more complex than single gene disorders. Polygenic traits involve intricate gene-environment interactions. Editing for enhancement rather than disease prevention remains scientifically uncertain. Nevertheless, the mere possibility raises profound ethical considerations about autonomy, consent, and societal pressure. Military applications introduce another dimension. Discussions about enhanced endurance, resistance to disease, or cognitive augmentation intersect with defense research interests. The concept of super soldiers created in laboratories captures public imagination, but the biological reality is constrained by trade-offs. Enhancing one physiological pathway often influences others unpredictably. Biology is not modular like software. Genes interact in complex regulatory networks. Large-scale modifications risk unintended consequences that may not manifest immediately. Parallel to CRISPR, mRNA technology has gained global attention. Messenger RNA serves as the intermediary between DNA and protein synthesis. By introducing synthetic mRNA into cells, scientists can instruct them to produce specific proteins temporarily. This approach does not alter the genome itself, but leverages cellular machinery for therapeutic effect. mRNA-based vaccines demonstrated the speed at which genetic instructions can be designed and deployed in response to emerging pathogens. Beyond vaccines, mRNA platforms are being explored for cancer immunotherapy and regenerative medicine. The flexibility of mRNA technology suggests a future where treatments are digitally designed and biologically executed. Longevity research has expanded into a multidisciplinary field. Scientists investigate caloric restriction pathways, senolytic drugs targeting senescent cells, mitochondrial optimization, and gene editing strategies to enhance DNA repair mechanisms. Aging is increasingly viewed not as an inevitable decline, but as a modifiable biological process. However, extending lifespan without preserving health span would create new societal challenges. Healthcare systems, pension structures, and economic productivity models are built around current life expectancy assumptions. Radical life extension would require structural adaptation across institutions. Public discourse often frames gene editing as playing God. This phrase reflects discomfort with altering foundational aspects of human biology. Yet medicine has always intervened in natural processes. Vaccination, organ transplantation, and antibiotics all modify survival trajectories. The distinction may lie in permanence and heritability. Editing somatic cells to treat disease affects an individual. Editing germline cells influences future generations without their consent. This temporal ethical extension complicates regulatory decisions. International governance remains fragmented. Some countries impose strict prohibitions on germline editing. Others permit research under controlled conditions. The absence of unified global standards raises the possibility of regulatory arbitrage, where individuals travel to jurisdictions with looser restrictions. Biotechnology tourism could mirror historical patterns seen in other controversial medical procedures. The challenge lies in balancing innovation with precaution. From a technical perspective, CRISPR is not flawless. Off-target effects, where unintended DNA segments are cut, remain a concern. Even low-frequency errors could have significant consequences in clinical contexts. Researchers are engineering next-generation Cas enzymes with improved specificity and exploring alternative editing systems that modify DNA without creating double-strand breaks. Base editing and prime editing represent refinements designed to reduce collateral damage. Each iteration moves gene editing closer to clinical reliability. The societal narrative around human upgrades intersects with cultural identity. Concepts of normalcy, disability, and diversity become contentious. If certain traits are editable, does society pressure individuals to conform to perceived genetic ideals? Disability advocates caution against framing genetic variation solely as pathology. Diversity has intrinsic value beyond biomedical optimization. The power to edit genes must be contextualized within social ethics. Aging research may ultimately reveal that biological decline is multifactorial and resistant to single interventions. Telomere extension alone will not halt systemic aging. Epigenetic reprogramming without genomic stability safeguards could introduce cancer risk. True longevity breakthroughs likely require integrated approaches combining gene editing, metabolic regulation, immune modulation, and environmental optimization. The path is complex but scientifically grounded. Economic implications are profound. Biotechnology startups attract substantial venture capital. Pharmaceutical companies invest heavily in gene therapy pipelines. Intellectual property disputes over CRISPR patents have highlighted the commercial stakes. The integration of artificial intelligence into genomics accelerates target identification and drug design. Machine learning models analyze vast genomic datasets to identify correlations between genetic variants and disease risk. The convergence of AI and gene editing may redefine personalized medicine. Cultural perception evolves alongside capability. What once seemed ethically unthinkable may become normalized as safety improves. In vitro fertilization initially faced opposition yet is now widely accepted. Gene editing could follow a similar trajectory if therapeutic benefits are demonstrated convincingly. The boundary between treatment and enhancement, however, remains contested territory. The question, can we remove aging, encapsulates both aspiration and uncertainty. Biological aging is not governed by a single gene switch. It emerges from cumulative cellular damage, regulatory drift, and metabolic constraints. Partial interventions may extend healthy lifespan modestly. Radical life extension remains speculative, yet incremental progress compounds over decades. A 10% increase in average health span could transform public health outcomes significantly. Ultimately, CRISPR and gene editing technologies place humanity at an inflection point. The tools to modify life at its source are real, operational, and advancing. Cas9 enables targeted genetic alteration. Telomere research illuminates mechanisms of cellular aging. Epigenetics reveals layers of gene regulation beyond sequence. mRNA technology demonstrates programmable therapeutic delivery. Together, they constitute a biotechnology platform with transformative potential. The ethical debate will not be resolved by science alone. It requires philosophical, legal, and societal deliberation. Scientific capability expands continuously. Governance frameworks must adapt correspondingly. The challenge is ensuring that innovation proceeds responsibly, equitably, and transparently. Humanity has always sought to transcend biological limits. From agriculture to antibiotics, each breakthrough redefines survival boundaries. Gene editing represents a deeper intervention modifying the blueprint itself. Whether this becomes a tool for alleviating suffering or a catalyst for inequality depends on decisions made now. The science is powerful, precise, and accelerating. The question is not whether we can edit life, but how we choose to use that power.